Hi, and welcome to Chapter 3. So this week we are learning about the cell and its structure, its functions. And so let's begin with question number 20 about just what is the basic unit of life. The basic unit of life is a cell. Okay, so now we're going to learn about what is a cell, what makes up a cell. Um, Okay, so that's the answer to the first question. The second question, its major parts. So I'm going to list them first, and then we'll take a picture or we'll take a look at a picture and talk a little bit about it. So there are three parts to the cell. The nucleus contains the DNA, the cell membrane, which is the boundary on the outside, and then everything in between, which is called cytoplasm. All right, so let me show you a nice image here from the textbook. And remember that you do have access to these images, and you can, make them as big or small as you like. So here's the cell nucleus. And remember, we do have an atomic nucleus too, right? So just to distinguish, but whenever I say nucleus, I typically mean the cell nucleus. All right, so inside is located our DNA, okay, our genetic material. RNA is located inside and out. They call it, the quote unquote brain of the cell, controls a lot of activities such as protein synthesis or making proteins. There's a structure inside called the nucleolus, which makes ribosomes. Um, Ribosomes are made of RNA. RNA is part of that ribosome. All right, and that's the nucleus. Okay, then we have the cell membrane, sometimes called the plasma membrane, depending on what resource you're looking at. All right, so it's the boundary, it's protection, and then everything in between the nucleus and the cell membrane is called the cytoplasm, right? And that includes all these different organelles in here, each with different functions. So we'll talk a little bit about some of these organelles, and you do have to know kind of the, to be able to identify and then its basic function of some of the major organelles. All right, we'll take a look at that. Okay, but before we get to that, let's talk a little bit about that membrane, the cell membrane. And maybe I'll put here plasma or cell membrane, okay? Because again, both are interchangeable. So I'm going to describe a few things and then I'll bring up a picture so we can look more closely. I mean, here on this picture above, it's a line, right? But obviously there must be more to it. Because if we're made up of cells, and all living things are made of cells, and it's the cell membrane that separates the cell from other cells and other structures within the body, then there must be something kind of unique, something special about it. And it certainly is. So let me bring up a bunch of information about it before we take a look. All right, so the type of lipid that makes up a membrane is a phospholipid. And the majority of the cell membrane is made up of phospholipids. And if you recall, we talked a little bit about phospholipids when we talked about lipids. There's two ends to it. Remember, there's a hydrophilic head end, which has phosphate in it, and then the tails are hydrophobic, right? And what we also call non-polar. But those are the tail ends. And if we take a whole bunch of phospholipids and throw them in water, the head and the tails orient themselves to so that the heads are all facing water and the tails are all not facing water, right? And so it makes this double membrane or this double layer, which forms the membrane. Now, included in that double layer of phospholipids, there are some other things in there. We have proteins that are embedded in the membrane, and they serve different functions Some that go all the way through the membrane can act as channels through which materials can go in and out. And then sometimes proteins have little markers on the outside that help us to distinguish, our own cells versus foreign cells. All right. Some proteins have carbohydrate components that reach to the outside, and those are also markers and also receptors. Now, receptor is something that receives information or receives a molecule. So if we have a receptor on the outside and we have a molecule that can bind to it, then it can cause some kind of action inside of the cell to happen. And then lastly, there's cholesterol. So cholesterol is also a lipid, but and in the past it's kind of gotten a bad rap, but cholesterol is a necessary component to membranes. All right, so let me bring up a membrane that it's not just a little line. All right, so this is a cell membrane or a plasma membrane. Let me make it bigger so we can see it. It really is quite a fascinating structure, especially, when you think of, it contacts the outside world. It interacts with other cells and fluids and things like that. And how do we control, is it like a wall where nothing can go in and out? Well, no, it's actually very what we call fluid. So this is a model that's sometimes referred to as a fluid mosaic model of a cell membrane. And what that means is that a mosaic meaning it has lots of components to it, and fluid meaning that it's not like a brick wall or something like that. All right, it can move, things can move in and out and through it, along it, and it can actually change shape. So this is what it looks like. Some of these proteins act as channels. Okay, some of them are, hold markers on the outside. Okay, carbohydrate chains that stick on the outside that can act as markers or receptors. And when I say markers, I mean like, a self cell versus a foreign cell. So all my cells have self markers that tell the the rest of the body that, hey, I'm me. But when you put another cell into somebody else's body, or into my body, put somebody else's cells, their markers are foreign to me. And so I would see that as a foreign invader, and my immune system would be called up to destroy it and get rid of it, right? So those are very important. And so that's what like organ rejection is. It's putting somebody else's cells with these different markers in somebody else's body, and the body saying, that's not me. And so my immune system is going to attack it. And that's why a lot of recipients of organs and things like that, they have to take immunosuppressants to suppress their immune system from attacking that foreign material. Okay. One thing, maybe let's take a quick look. All right, so, if we have our cell membrane, and one way it can be fluid, so here's a nucleus and there's lots of organelles inside of it, but there are some of our cells that, let's say we have some kind of foreign invader here, bacteria. Some of our cells can eat it. We have some cells that act as what we call phagocytes. They're eaters. And notice how the membrane changes and allows for that bacteria to be taken into the cell like this. And then we can destroy it. We have other organelles called lysosomes that can come over and secrete its contents empty its contents into this new little vesicle, and then destroy the bacteria. But notice how the membrane is fluid. All right, it's able to extend and wrap around it. So if you think of like an amoeba is a single-celled organism, and it can extend its pseudopods, they call it, these little feet-like structures that can come out and then engulf things and eat things. So that's the same concept. Is the cell membrane, the plasma membrane is very fluid in that way. All right, so hope you have a new respect for our membranes and what they have to do, what they can do. All right, the next one, which kind of leads us to what I started talking about, which is, all right, now we know what the membrane kind of looks like. How do we, is it the same way that material is brought into the cell, or the same way that things are taken out of the cell, right? Because we have to secrete stuff. We secrete hormones and other substances like into the blood, and we have to make it inside the cell, and then we get rid of it. Okay, we release the hormone into the blood, and then it does some kind of activity. All right. And also sometimes we need to take things into the cell, such as that bacteria, or I don't know, building blocks for something, to make something. All right, so how do we bring things in and out? There are two kind of overviews, two mechanisms that are very general. One is called a passive mechanism, and the other one is active. Passive sounds just like it sounds, which is it doesn't take energy. If you're a passive person, in a fight, you're very passive, means you don't take action. Okay, so a passive mechanism would be one that doesn't require any energy. Now things happen, but no energy is required. Okay, so let's take a look at that first. So what are some examples of some passive mechanisms? The first one is, or the first type of passive mechanism is called diffusion. And let me define that. So diffusion is the movement of molecules from where there's a lot, a high concentration, to where there's less, at a lower concentration, until it's equally distributed. So an example would be if someone puts on perfume on their body right before they get into a car or something. So I put on perfume, I get in the car, close the door. Where is there more perfume molecules? Okay, it's right on my body. Where is there less? Okay, everywhere else in the car. But if I sit there, what happens is a passive process. And this is just, it's a process that governs all, molecules. So once I get in the car, the molecules begin moving from where there's more to where there's less, and it diffuses across. So the person right next to you is going to smell it first. I could use the example of someone farting or something like that, but let's use perfume. So, but molecules will go from where there's more, and then they begin what we call diffusing. They begin moving to where there's less. And again, it's a natural process. They start moving, and eventually, if you sit there long enough, or if I sat there long enough, those perfume molecules would be equally distributed within that car. All right, and then who would smell it last? It'd be the person farther away. All right, I think most people kind of understand that concept. Here's a picture of an example. I gave the example of perfume, but if we look at an example of a block of, is that a sugar cube or something? You put a sugar cube inside of a water glass, okay, a beaker of water, and it begins diffusing. It will move from where there's more to where there's less. And there are forces kind of pushing it in that way. There's something called Brownian motion where all molecules are in constant motion. So if you have a whole bunch sitting together, they're going to hit each other and move away from each other. And the more concentrated it is, the more it's going to hit things and move farther away. The lower the concentration, the slower that will take. But anyway, so then it gets completely filled with those sugar molecules, right, over time. The next type of passive movement is a special type of diffusion. This special type of diffusion is called osmosis. Osmosis is specifically diffusion of water, okay? And you know, water is such an important part to living things, so we have to talk about what kind of laws govern water movement. So osmosis is diffusion of water, okay? So we're looking from where there's more water to where there's less, but it has to cross a membrane. And I call it a selectively permeable membrane. All membranes are selectively permeable in that it will allow some things to pass, but not everything, right? So in this case, or in the case of cell membranes, water passes easily across membranes, okay? But larger molecules, what we call solutes, like sugar and salt, those things are too big to pass easily across the membrane. So they stay on one side, but the water can move back and forth. So let's take an example of where you might see this in real life. Okay, so let's say in a hospital, patients are given IVs. Okay, fluids are put into the veins and to either distribute or you know, give certain drugs that are necessary, or maybe just to hydrate someone who's dehydrated or something like that. So the concentration of that solution that's being put into the body is important. All right, because we know that water moves across, and it moves across freely based on the concentration of that water and the concentration of the solutes that are dissolved in it. Okay, so in a red blood cell, maybe I'll show a picture first. So we're going to look at the example of a red blood cell. Okay, and we're going to take a look at, and then I'm going to describe kind of what this looks like. All right, so here is, I'm going to make it a little bigger. Here's red blood cell, and this is a normal red blood cell. And if we expose it to 3 different solutions, okay, so they're not all the same. So if we put a red blood cell in water, 100% water, no solutes, nothing dissolved in it, okay, is the cell happy or is it not happy? It actually is not happy. Water that is 100% on the outside will move into the cell because there's less water on the inside of the cell. So inside a cell, intracellular fluid is, sure, it has water in it, but there's also solutes, dissolved solutes. Okay, there's salt. It's a little salty solution inside, not huge, but there's a slightly salty solution on the inside of our cells. And so because of that, water is going to move, all right, from high to low concentration. One way I like to think about it is water always moves toward where there is more solute, okay, where there's more salt or sugar. So you have to just determine where is there more salt or sugar on you know, one side of the membrane, and wherever there's more salt or sugar, the water is going to go toward that. All right, so in this case, there's more salt inside the cell, so the water will go inside the cell. Okay, so even though it's water moving from high to low concentration, because the salt doesn't go anywhere, it stays inside the cell, but the water just goes, I like to think of it as to dilute the salt or the sugar. Over here on this side, this solution has more salt or sugar on the outside. And I know that because water is being pulled out of the cell. And water will go from where there's more water on the inside to where there's less water or more salt or sugar. Okay, maybe I should, okay, I'll draw that in just a moment. We'll talk a little bit more. And then here, this is what a normal red blood cell looks like. It's what we call a biconcave disc. So it's like a donut with the center not quite punched out. That's the shape of a regular red blood cell. And so here in this case, it looks like water's moving out, but an equal amount of water moves in. So the concentration of solution is the same inside and outside. So we don't get any net movement of water into or out of the cell. It stays about the same. All right. Let me talk really briefly about, let me give an example. And okay. All right, so let's take a look at our little red blood cell. So here's a red blood cell. And here's a solution it's sitting in. So this is some kind of solution with water. Now, one thing I want to point out is, you know, when we have a solution of something, okay, if I make like a sugar solution and it's a 10% sugar solution, that means I put 10 grams of sugar. Let's make sugar ring. 10 grams of sugar in 100 milliliters of water. Okay, and so so we have, I'm just gonna do it like this to make it easier to understand, 90% water and 10% sugar. Okay. And so now let's say I want to look and compare that to a solution that is a 40% sugar. So I'm going to make this much bigger. Oh, let's do that. 40% sugar. And that leaves only 60% water. All right, so you can see where as you increase the solute percentage or the concentration, you decrease the amount of water in it. All right, so do you see that? The difference between a 10% sucrose and a 40% sucrose. So you put 40 grams of sugar and you dissolve it into 100 milliliters of water. And this is kind of what it maybe looks like if we were to like separate it out and stuff. All right, so now we have our red blood cell. And I will tell you that inside the red blood cell, oh, I should have made this salt. Okay, it's sugar or salt, NaCl is salt. Okay, but the concentration or the concept is the same. So inside red blood cells, I remember I mentioned that it is a little salty. So it is less than 1% NaCl. So a little bit of salty, but it's actually 0.9%, 0.9% NaCl is inside the red blood cell. Less than 1%. Well, that would mean that like 99% is water, right? So let's say 1% versus 99%. What if I put it in a 0.9% less than 1% solution on the outside. In other words, it's the same solution inside and out. Same is iso. So I would call this an isotonic solution. Iso means the same. All right. And so what would happen to our red blood cell? And we saw nothing. It's happy there. Okay, it's not distorted. You don't have a lot of water coming in or leaving, maybe a little bit coming in and going out, but it would be about the same. So isotonic solution is the same. Now, what if we put the red blood cell into a solution? And we know the solution inside is always the same. So it is a 0.9%, 0.9% NaCl. All right, so here, and this is always the same, but let's say I drop it into a 40% salt. Okay, a lot of salt. Very salty solution out here. But inside, it's 1% or less than 1%. Which way does water flow? And this is a very important concept in terms of, like IVs and tissues and things like that. So remember that water always goes toward where there's more solute. So water goes toward where it's saltier or more sugary. And I always think of it like, if it's sugary over there, all right, my water's going to go to dilute it. So inside is not very salty, only less than 1% salt. This is 40%, which means that water is going to go toward the salty solution. It's going to pull it out. So we call this solution on the outside, this solution is called a hypertonic solution. Hyper means more. Oops, hold on. Go back. Okay, hyper means more. So this is more tone. In other words, there's more molecules that press against something and gives it like tone to it. So this has more salt in that solution. All right, so hyper means more. All right, now what if I put that little red blood cell into a solution that has less salt? So let's say it has 0 salt. No salt. But there's a slightly salty solution on the inside, right? Oops, sorry. Okay, so no salt, which means it's 100% water. Okay, 100% water on the outside, no salt. And there's our little red blood cell with its less than 1% salt. Where is it more salty? It's more salty inside the cell, right? So it's saltier there, so water flows into the cell. And this solution out here is called a hypotonic solution. Hypo means less than. Right? We saw hypochondriac, it means less or under the ribs. Hypothermia is your body temperature goes below normal. Right? So hypo means less. So what's going to happen to these little red blood cells? If it's in a hypotonic solution, the water moves in and that red blood cell swells and can even burst. So this is called swell and even lyse. Lysing is burst. What happens to this red blood cell? Nothing, it's happy. Okay. If a red blood cell can be happy. And then this third one, what happens to it? Oh, we see the water coming out, being sucked out. All right, it gets all weird like that. and it shrivels. And another name for shrivel is crenate. Crenation is like something shrinking or shriveling. All right, with red blood cells. All right, does that make sense? Okay, so maybe I'll do this so you can get a whole picture. Okay, very good. So let's go back now and take a look and fill this in. All right. So I hope this makes sense now, what the shape of the red blood cells are going to be like. And let me see if I can. All right. And I think that's good. Maybe I'll show, well, this is a little backwards. So I won't show this picture. And then when you actually look at red blood cells under the microscope and you add different solution concentrations, then you can see them like getting all fat or shriveling up. So it does, it is apparent under the microscope. So what does this mean like for, let's say you're in surgery or you're a healthcare person in surgery and they're doing a biopsy and they take a sample of tissue out of the liver. They're seeing if it's cancerous or something, and they cut a piece of liver off. They put it in a little dish and they give it to you and they say, all right, keep it moist. Don't let it dry out and take it down to the lab. All right. And you go, okay, I got to keep it moist. And then you go and you put water on it. Okay, I'm going to just put some water on it to keep it moist. All right. And then as you take it down to the lab, are they going to be happy about that? No, because adding water is a hypotonic solution. That means water is going to move into the cells and distort the cells. What you should do is put an isotonic solution. So have you heard of normal saline or, 0.9% NaCl? Okay, normal saline is 0.9% NaCl. It's an isotonic solution, a little bit of salt. Okay, so you want to put it in an isotonic solution. All right, very good. So now let's take a look at cell structures and functions. So small little organs are called organelles. So an organelle are little organs. So of course, these are located within the little structures within a cell. And like your organs that are all different and perform different functions, so are the organelles. Each one looks different, each one has a different function, but together they contribute to the overall functioning of the cell. All right, so we have lots of different organelles in the body. And, actually, let's finish this one. I never finished it. I just talked about diffusion and osmosis, and there are more. Sorry about that. I got distracted. All right, so let's take a peek. We've talked about two types of passive mechanisms, diffusion, and the other one is osmosis. All right, there is a third type of passive movement of molecules. This is called facilitated diffusion. So let me get that here. It's not A, it would be, it's one I. This one should be I. Uh-oh. Okay, that's not right. All right, so this is the third type of passive transport, facilitated. So this is, again, diffusion, so movement of molecules from high to low concentration, but it goes across the membrane, but it needs the help or the facilitation by a protein. Okay, so let me get a picture. Sure. So oftentimes these molecules are either very large, so they don't fit, okay, or just that they're not lipid soluble. They don't go through the membrane, and so they need a special protein channel in order to help facilitate diffusion. Okay, so it's facilitated, and if you're a facilitator, you go and you're not involved in maybe the dispute, but you help the process for it to be resolved. So same here, the protein isn't directly, one of the molecules going across, but it helps or it facilitates or aids in that process of diffusion. All right, so those were the passive movements, passive mechanisms. Next, we have the active mechanisms. Okay. All right, so the active mechanisms require energy. All right, so I hope that's clear. Now we're going to do B, the second type of mechanism, and these are the active ones, the ones that require energy. Okay, and energy in the form of ATP. Now there's different types of active mechanisms. The first one is actually called active transport. There's other types too. So this is specific. And moving, and it's not specific, it's general, and we'll look at some specific types. It just means moving air molecules against the gradient, well, it's going from low to high concentrations instead of high to low. So I think of passive mechanisms are like getting on an inner tube in a stream or a river, right? And you just lie there like a lazy river and you float down. Doesn't take any energy. But if you wanted to go upstream against the current, what we call against the gradient, then you would need to expend energy, right? You'd have to paddle, kick, whatever to get back up against the flow. And so active transport is moving against the flow. You go from low to high concentration. And so this isn't, a normal passive thing. It's active. You need energy. So let's look at some other mechanisms. These are a little bit more specific. This is the first one's called endocytosis. So endo means within. So this is a cell bringing something in to the cell, and it does it by way of vesicles forming. I showed you, gave you an example of bringing a bacteria in. So this is just another picture kind of that. Oh, maybe I should. I'm going to put the next one. Okay, but I'm going to add a picture here. So this would be an example of endocytosis, right? We have molecules out here. We want to bring it in. And so the membrane curves in and brings it inside, right? The other type is called exocytosis. Exocytosis is the opposite. Okay, it is taking substances taking substances out of the cell. Okay, so one's bringing it in, one's taking it out, and just imagine it being kind of the opposite. So remember that the membrane is very flexible and it can join with the membrane to release the substances. Okay. Okay, I think that's it. So one group is passive and one group is active. All right, now let's get to our little organs. So I'm going to bring this up again. And let's take a look at different organelles. So maybe I'll put the, I I'm going to put a picture here first. And then we'll go through and look at some of the organelles. Okay. So here's our little cell. Okay. And I'm not going to have you know everything, but let me bring up a bunch of structures that you are responsible for. The first one is the nucleus, and I mentioned it before. So we're going to look at the different types of organelles. Nucleus, one of the largest, contains genetic material, the DNA, and acts as a control center for the cell. The next one is the endoplasmic reticulum, and there are two types of endoplasmic reticulum, and based on the way it looks. Okay, one is kind of bumpy, and it looks kind of rough, and the other one is much more smooth. There, oops. Why did sometimes just getting it. Okay, I'll have to do that. All right, so let's start with the rough endoplasmic reticulum. It is continuous with the nuclear membrane. So surrounding the nucleus is a membrane. It too is a, just like all the other, just like the other membranes. And so, all right. So it's a bunch of like tubes that is continuous with the nuclear membrane, and it has ribosomes on the outside. So it's kind of studded with ribosomes. And so here we have the nucleus. And it's kind of hard to see. And so we have all these tubules here. Okay, see there's ribosomes on the outside here. So this is rough endoplasmic reticulum. And they're just transport passageways to get material into and out of the nucleus, or just to transport things around. All right, so if it's got the ribosomes on the outside, then it's a rough. If it doesn't, like over here, there's no ribosomes on it, this is called smooth endoplasmic reticulum. All right, let me see. Okay, there we go. Continuous. Okay, I was just making extra copies of things. I want that. Okay. So the smooth ER is where fatty acids, some steroids are produced, and it can detoxify drugs and inactivate drugs and other things that are potentially harmful. Okay. Next, we have the Golgi complex, sometimes called the Golgi apparatus. So Golgi is okay. Okay. So there's the Golgi. These are like little flattened pancakes, but they're sacks, empty sacks, and they receive molecules from the endoplasmic reticulum, and then they alter them. So, and then they transport them to other places in the body. So it modifies the molecules from the rough endoplasmic reticulum, packages it up, modifies it if it needs it, and then transports it to other places. So the Golgi, right here, this purple one. All right, so these are like flattened sacs, but they do form vesicles that can kind of blub off of it, and that houses maybe a protein or something that is transported outside of the body, okay, by exocytosis. So that's the Golgi. Okay, let's see what's next. The lysosome. The lysosome, and I've mentioned this really briefly, is another organelle that that contains digestive enzymes. So it's kind of the digestive system of the cell. All right, it's got 60 different enzymes in there. And so whatever it fuses with, it can break it down. So let's say that you enclose a bacteria, you can digest it by putting enzymes in there and making it die or go away. But other things too. So sometimes cells die and you want to get rid of the debris in the body. You could do that through lysosomes also. All right, next we have the mitochondria. Mitochondria, sometimes called the powerhouse of the cell. Okay. It's at the mitochondria that food, or within the cell, that food that was prepared then go to the mitochondria where we produce ATP, so energy. All right, but it takes the food we eat, and that needs to be modified and then taken to the mitochondria where we make ATP from that. All right, and so that's a whole process of cellular respiration, and something we won't talk too much about here. We might mention it though. All right, so the last thing are the ribosomes, which I've already mentioned. Ribosomes are attached to the rough endoplasmic reticulum. This is where proteins are made, protein synthesis, and it's made up of RNA. Okay, and this is where we make proteins there in the cell. All right, so those are the ones. Of course, there's more, but these are the main ones that you will be responsible for. I may quiz you on, so be sure to take a look at that. Okay, what's next? Now we're going to really kind of switch gears a little bit and look at something called gene expression. All right. So, and then a couple processes called transcription and translation. So let me define that, and then we'll talk about that. And really, this is about making proteins. How do we make a protein, right? How can we make a hormone? How do we build our muscles, right? We must be making proteins in our muscle cells that make them bigger, right? So, we're making proteins all the time. The process of gene expression is just control over what proteins you're going to make. Okay. So, let me bring up a picture here. And let's look at the cell nucleus really quickly. All right, in the cell nucleus, we have DNA, and we've gone over a little bit about the structure of DNA, right? It's a double helix, we call it, 2 strands of nucleotides that are joined together by the nitrogen-containing bases. Okay, and the way DNA is situated in our cell, it's wound around proteins called histones, and it's just packed. It allows us to pack better within the nucleus. Now, one thing that happens is there is a point in the cell life cycle where the DNA is going to really coil up tightly to form structures called chromosomes. All right, and so these are chromosomes here. That's what that is. And then here's the DNA. Okay, with that, how do we make a protein? Maybe I'll draw that out, and then I'll show you a picture. Okay, so let's say we have our cell here. Here's our cell, and in the cell we have a nucleus, and I'm drawing little openings because there are little pores that are located in the nuclear membrane. All right, now inside the nucleus, we have the DNA, and it is double-stranded like this. All right. Now, pieces of DNA form what we call genes. A gene is a piece of DNA that makes a particular protein. So I might have, the eye color gene, okay? And so that gene, that specific group of nucleotides will be the blueprint for making one particular protein that, let's say, gives my eye color, a certain color, right? And we inherit that from mom and dad and, end up with, a certain color eye. Well, that protein that you made was directed by a gene, which is a piece of DNA. So let's say this particular piece of DNA, this gene, is a gene for, let's say, eye color. It's not that simple, but let's just say that. All right, so how do we get from a nucleotide or a group of nucleotides in your nucleus out here to a protein? Because that's where we want to end up with. We want to end up with a protein. Okay, so it's through several processes. The first process is called transcription. So transcription is this double-stranded piece of DNA is made into an RNA, or we make a copy, an RNA copy from that DNA. So what that looks like is if, here's our DNA, and let's say the gene we want is right, let's say here. So what happens is this DNA actually will unwind at that spot. Okay. Oops. And it unwinds right at that spot. And so the DNA then, or from the DNA, we make a copy. So this copy would copy the order of nucleotides, the adenine, thymine, guanine, cytosine. So whatever the pattern is, we make a copy of RNA. This is called a messenger RNA. This is the first type of RNA. All right, which has the code of that gene. And then strangely, this DNA winds back up after that, that piece of DNA is read and made a copy of. This first process is called transcription. Okay, we transcribed it. So if you look at it like a court of law, there's a person who's typing, they're the transcriptionist. They transcribe. So what are they doing? They're taking what's said in court, all the oral part, the what people are saying, and they put it into a different form, but the language is the same. So they're not going to put it in a different language. They're going to put it, let's say here in the United States, in English. but they're changing the form of it. It's going from an oral language to a written language. All right. That's what's happening here. We don't change the language. It's still language of nucleotides because the RNA is made up of nucleotides also. But it changed, what did change was the format. So what was once double-stranded DNA is now converted into a single strand of RNA. But it contains the same information, and it didn't change the language. We're still in the language of nucleotides. All right, so what happens to this little message? So now we have a little message, a messenger RNA that's going to leave the nucleus and go to a ribosome. I'm going to greatly enlarge that ribosome. So the ribosome here is actually would be like attached to the endoplasmic reticulum. And this little message is read by the ribosomes. Now floating around the cell, we have other RNA called transfer RNA. Okay, a single-stranded nucleotide. But now this RNA is attached to an amino acid. All right. So, and depending on what the code is here, it will bring different transfer RNAs because there's lots of different amino acids. So let's say this is amino acid number 1, but then we have another transfer RNA here that has attached to it amino acid number 5. And there's like 20 different types of amino acids in living things. All right. So let's say we have all different kinds. And so whatever the code is here in the RNA, the proper transfer RNA that matches that code is going to bind here and bring its amino acid. So let's say this is amino acid number 1. But then, depending on what the next set of nucleotides are, another transfer RNA will come and bring amino acid number 5, right, etc. And we're going to read the message until we get all a bunch of amino acids, and we join the amino acids into a strand, which then will form eventually a protein. Okay, this process is called, so this process here was number 1, transcription, and then the second process is translation. So let's take a look. Now we have our messenger RNA at the ribosome. The message is read, and we bring different transfer RNAs with their associated amino acid. Okay, and so if this comes up and binds, it will bring its amino acid. Then we have another transfer RNA that will bring another amino acid 5. Then we have another one that brings amino acid number 10. All right, and then we join the amino acids together, which then will form a protein because amino acids are the building block, right? And so this process is called translation. Now, when you translate or you use a translator, what is that person doing? It's changing the language, all right? A translator changes the language. And what did we do here? We went from a language of nucleotides and we changed it to the language of amino acids, which is the protein language. All right, and that's what the difference between transcription and translation is. But the whole process of transcription and translation is to make a protein. So let's say you start working out. I decide I'm going to lift weights now. So I start lifting weights. What's going to happen to my muscles? They enlarge. Well, wow, what happened? How did we get from working out to muscle protein? Okay, because we have to make protein muscle protein in order to build our muscles, right? We can't just make it out of nothing. So what happens here is as we work out, we stimulate the proper muscle gene. To unwind, we make a little messenger RNA of that muscle protein, and that will go into the cytoplasm where it will bind to ribosome, and the proper amino acids will come in and join together to form a protein. Okay, all right. So let's go ahead and I hope that makes sense and go back. We're almost done here. Okay, so transcription, conversion of DNA into messenger RNA, and translation is conversion of the information in the messenger RNA into protein. And that's all you have to know, right? I gave a lot of detail just so you can understand it, but not necessarily that you have to, recreate all that. So this is a kind of a picture of what I was showing as the information passes along. And here's the ribosome that's reading the messenger RNA. All right. Let's take a look at the cell cycle. Okay. Okay, so let's take a look at a cell and kind of its life. That's what the cell cycle is. It's kind of its life cycle. So just like us, we have, oh, there's one more thing I wanted to add. Okay, let me add something to this other picture here. I have a couple other pictures to add that you might like. All right, so this is kind of showing that. And then this is that process of making a message, the messenger RNA here. All right, so it unwinds the gene we want, and then we make a little messenger RNA here. And then it's released. This winds back up, and it goes along its way. So, oh, and then to kind of as a, maybe this might help to summarize kind of this process. And we call it sometimes the central dogma of protein synthesis. Not quite sure where that comes from, but this is the process going from the DNA in the nucleus to a protein. All right, so now this next one, the cell cycle. It's the life of a cell, right? So if we take a look at like our life, we spend a certain amount of time as an infant, as a child, as an adolescent, as an adult, as an old person, an elderly person, right? And if we were to make a little pie chart, we could spend, put it in how much time we spend in each one of those stages in our life. So I'm going to draw what's called the cell cycle, and it is a pie, kind of a pie. Oh my gosh, that looks so bad. Okay, anyway, so there's my pie. And we spend some time in different phases in the life of a cell. So let's say we have our little cell here. Here's a nucleus. Okay, our DNA in there. And it's going to go through different phases. 3 to be exact. The first one is called G1 phase. This is a growth phase. Okay, the cell grows, we make more organelles and things like that. The second phase is called the S phase. The S phase is DNA synthesis or replication. We make an extra copy of DNA because what we're doing is preparing for division. The cell is going to divide, and we're preparing for that. So the S phase is we have to make an extra copy of DNA, because if we didn't, whenever the cell divided, it would have to split the DNA into half. so that each cell got some DNA. And every time it divided, it would continue to divide and split the amount of DNA in half and half and half until we'd almost have no DNA in the cells that were being divided. Okay, so instead, we make an extra copy of DNA. So by the time we divide the cell, each cell will get a full copy, right? Does that make sense? And then the last phase is called the G2 phase. And this also is growth as it prepares to divide. Okay, now these three phases together are called interphase. Okay, interphase is made up of G1, S, and G2 phases. Oops, G2 phases. Okay, now a cell will spend most of its life in interphase. And then we come upon the process of dividing. Now, the process of dividing is complex, and it's actually made up of two parts. The first part is called mitosis. Mitosis is dividing the nucleus. Okay, so mitosis is division of the nucleus resulting in two nuclei that are identical to the original. We're making clones. All right, so mitosis is just dividing the nucleus. And then the second part is called cytokinesis. I see the word cyto like cytoplasm. So cytokinesis is division of the rest of the cell or division of the cytoplasm. All right, so mitosis and then cytokinesis. And in fact, mitosis and cytokinesis kind of happen at the same time. As the nucleus is dividing, then the rest of the cell begins to divide. Mitosis can be broken up into 4 different phases. And I'm going to label them PMAT. Okay, so PMAT. These are prophase. So we're looking at mitosis. We have prophase, metaphase, anaphase, and telophase. And there's very specific things that happen in each one of those phases with the DNA, because the DNA is what's in the nucleus and what we need to divide, right? All right, so let's go back. And go to our copy here. Okay, and I'm going to write out the 3 phases of interphase first. Okay, G1, S, G2. Then I'm going to put the cell cycle in. Okay. All right, so here it is, G1, S, G2. There is, or they did a little differently. These are the 4 phases of mitosis, and then they just say cytokinesis occurs. All right. So what does this look like? Actually, let's go to the end. We're going to answer these last questions. Mitosis, cytokinesis, and what is happening in each one of those phases. So I'm going to put that in here as I've defined it already. Mitosis is division of the cell nucleus, resulting in two nuclei identical to the original. Cytokinesis is division of cytoplasm. These are the 4 phases of mitosis. Okay, and I'm going to now show you what happens. Maybe I'll just tell you first. In prophase, we can just see the chromosomes because before that, before mitosis occurs, and when it's in interphase, we can't really see individual chromosomes. The DNA is kind of all loose like that. But as soon as we go into prophase, it begins coiling up. The DNA begins coiling really tightly and forming these structures called chromosomes. And I do put here the nuclear envelope disappears. So the membrane around the nucleus just kind of goes away and it will reappear. Metaphase, all the chromosomes are going to line up in a single file line. It's crazy. Anaphase, all right, because the chromosomes are already duplicated, now we just split it apart so that each side or each cell will get a copy of all the DNA. Okay, and telophase is the chromosomes move to the opposite sides of the cell, and then we split the cell in half. So let's take a look at what that looks like. Okay, this is from your textbook. Okay, so let's take a look. All right, we're going to start with prophase, or just here in interphase. We can't really see individual chromosomes. Let me see if I can. All right, it's just kind of, we can start to see kind of squiggly stuff, but we can't really actually see individual chromosomes. Prophase, they look like worms. The worms appear, okay? But it's chromosomes, so we can start to see the chromosomes. Now, humans have 46 chromosomes, or 23 pairs of chromosomes. 46 total. Okay, so then they all become apparent. There should be 46 chromosomes in there if this is a human. But you know, like different organisms have different number of chromosomes. Corn has 10 chromosomes. All right, but that is one of the reasons why we can't have intercourse with and make viable children with other animals, right? Because our chromosome numbers are all different. And so we get weird numbers of chromosomes and offspring don't usually live. All right, so then metaphase, they all line up. Look they all line up right in the middle. Here they are lining up. This is an organism with only 4 chromosomes. Imagine us humans with 46 chromosomes lined up. Complicated. All right, anaphase, the chromosomes split apart because remember here, they're already duplicated. They're duplicated. And so now we can split them apart and the chromosomes go to each end in telophase and we can see cytokinesis happening. The cell is pinching inward. Okay, and then cytokinesis occurs. The cell membrane, the nuclear membrane reappears. And we get 2 individual cells that are identical to the original. Okay, and so now we have 2 cells with the same amount of chromosomes. So in this case, it would be each one would have 46 chromosomes. Because here, or in the S phase of interphase, before we even began mitosis, we doubled our chromosome amount. See how they're little X's? Well, an individual chromosome is like a single line. But we doubled it so that when we split it, each side got a copy. All right, now why is mitosis important? Okay, because we start out as a single cell. And how do we get from a single cell to a multicellular organism? It's because of mitosis. And we just make more cells. And so what does that tell you about your cells, the DNA in every one of your cells that have a nucleus? Red blood cells don't have nuclei, mature red blood cells. But anyway, so all the cells with your, with nuclei. They all have the same copy of DNA because it originated from a single nucleus, right? And so if you commit a crime and you leave a few cells at the crime scene, they have a full copy of your DNA because of mitosis. If you get a cut on your arm, okay, oh, and magically it heals. What is that? Oh, that's mitosis. Okay, cells are undergoing mitosis and making more copies. All right, what is cancer? Cancer is cells that have lost control over mitosis. So a normal cell that becomes abnormal, it begins undergoing mitosis too much. And you make too many cells, and most of the time those cells are not useful. Okay. And all they do is they crowd out tissue, they press on nerves and on vessels, and eventually, they take over. So this is important. Mitosis, this process, I would encourage you to spend a little time to really understand this because, it's a basis for a lot of important things in our body. It gets us to, where we are. All right, guys, thanks for listening. And I hope it's understandable. Okay. And don't forget to take the practice quiz, the chapter 3 practice quiz multiple times, not only until you get a perfect score, but take it a lot because then you get to see all the questions from this chapter that could appear on the first exam. Okay. All right. Thanks.